Hello again. In the second part of my video dedicated to Leishmania development and sandfly gut and transmission to the host, I will tell you something a little bit more about early phase of survival of the parasite inside of the sandfly gut. So, when enter the midgut, first amastigotes, they are actually faced with a hostile environment. So there are some natural barriers to Leishmania development inside of the gut and during the first day after blood feeding. And this barrier includes three major, let's say, processes. It's the secretion of the proteolytic enzymes, it's the peritrophic matrix which surrounds the ingested blood meal, and sandfly immune reaction. Proteolytic enzymes. So ingestion of blood meal actually induced the secretion of the digestive enzymes. Some earlier studies before, they described that parasites transforming from asmastigotes to promastigotes are susceptible to sandfly proteases. But recent experiments did not show any direct negative effects of these enzymes of transforming parasites. There is a significant level of the proteolytic activity detected 6 hours post-blood meal, and peak level are reached 18 to 48 hours after blood meal, depending on the sandfly species. There are several enzymes found inside of the sandfly gut, and these are major serine protease, trypsin, and chymotrypsin-like enzymes. There are also some other transcripts found for the other proteolytic enzymes like metallocarboxypeptidase, actin-like metalloproteases, alanine aminopeptidase, and novel serine protease, as well as some aminopeptidases. So in order to complete developmental cycle, Leishmania must have to develop mechanisms actually to overcome this hostile environment of the blood-fed midgut. And there are also results and research studies that show that parasites can actually manipulate the level and the timing of protease activity in the midgut. For example, Leishmania species can suppress or it can delay the peak of trypsin and aminopeptidase activity inside of the midgut. Leishmania by itself also possesses the mechanisms that increase the resistance to proteolytic attack without inhibiting the overall proteolytic activity in the midgut. These are known as phosphoglycans, and we have two major groups important for this topic. It's lipophosphogycans and proteophosphoglycans. So the fresh tissue amastigotes and fully developed promastigotes are relatively resistant, whereas the parasites within early stage amastigote to promastigote transition 2 to 8 hours old become highly susceptible to killing. What usually happens, and studies show that in most experiments around 50% of the parasites actually die inside of the peritrophic matrix. Now, another problem for the Leishmania inside of the midgut is actually this already mentioned peritrophic matrix proteinase and also development of chitinases. So the peritrophic matrix is actually an extracellular acellular chitin containing envelope, which is most in one insect separates the gut lumen from the midgut epithelium. It is composed from chitin, some proteins, and some glycoproteins. In Nematoceran diptera, including sandflies, females produce type 1 peritrophic matrix, which is secreted by the midgut epithelium in direct response to the dilatation of the midgut caused by blood feeding. 2-3 days after blood meal, the peritrophic matrix structure actually appears wrinkled and then starts to break down. At the end of the digestion process, sandfly chitinase disintegrate to peritrophic matrix on its anterior or sometimes find posterior end, not mostly anterior end. And in the beginning it was thought that both and Leishmania also actually secreted the chitinase and also involved in disintegration of the peritrophic matrix, but some studies also show that it is not necessary, basically because the sandflies already secrete enough chitinase to disintegrate the peritrophic matrix. Leishmania chitinase does not seem to participate very much in this process, according to some authors. And also there are some proteins and peptides involved in innate immunity. So sandflies, they have their own immunity, which actually prevents to be infected or to be damaged by some viruses, bacteria, or parasites. And this immune response plays a very important role in the control of bacteria, for example, and also some parasitic infections in blood-sucking insects. This group are also called and known as defensins. It's cationic antibacterial peptide, and they have been described also to exist in the fat body or in the side of the midgut. Transcripts that coding the several other putative components of the innate immune response have also been detected in scent-like midgut, and these are known as pattern recognition proteins, glycine-rich proteins, serpins, or antioxidant enzymes. Establishment of infection and attachment of the parasite to the midgut epithelium. So, as the blood digestion proceeds, parasites need to bind to the midgut epithelium to avoid to be excreted with the blood remnant. Following the escape from the endoperitrophic space from peritrophic matrix, the parasites actually attach themselves to the midgut, inserting the flagella between the epithelia and microvilli, and also this binding is prerequisite for the further development, because in that way they stay inside of the midgut. The midgut binding is stage dependent. It's being limited to the form found in the middle phase of development, like nectomonad forms and also leptomonad forms, but absent in procycling and metacycling. So only nectomonad and leptomonad attached, or nectomonad attached, they then transform to leptomonad, and then they detach and then go towards the stomatal valve. Procyclics and metacyclics, they don't attach at all. There is no obvious ultrastructure, let's say, modification of the flagellum associated find up to now with midgut binding. It remains unclear whatever exactly is the involvement of the flagellum in essential, let's say, or just a reflection of the fact that being at the anterior part and the flagellum will contact the epithelium first and fits between the microvilli. So we don't know exactly if it is necessary to get that attached with the flagellum or not. The binding disc mechanism differs between scent-fly species. For example, in Papataceae, the attachment to the midgut microvilli is controlled by galectin, which serves actually as a receptor for terminal galactose present in Leishmania major and Leishmania puranica, for example. It's lipophosphoglycan, the main surface glycoconjugate on parasite surface. More recently, there are found some others also, flagellal proteins like FLAG1 and SMP1, which actually demonstrated in Phlebotomus papataceae infected by Leishmania major. In contrast, in permissive scent flies, the attachment does not require the LPG, the lipophosphoglycans, as even the parasites deficient in lipophosphoglycans develop inside of the midgut. Thank you very much for watching the second part of this video, and I will continue soon.